Good morning students, I am Urmi Desai from Saraswati Academy Ratavapi. I am your new biology science teacher. So today we will start with your first chapter of the biology, that is chapter number 5, the fundamental unit of life. Okay, before starting the chapter, first we will understood that is what is biology. You must have heard about that is science is divided into three parts: chemistry, physics and biology. So basically biology is the study of living organisms. Biology is related with the study of living organisms. So your first chapter, that is the fundamental unit of life, how living organisms are made up of, your chapter number 5, that is how we consider any organism as a living. What are the structural or functional constituents which are present inside that organism, right? So let's start with the chapter. So in the chapter, the first basic concept we have to understand, that is the fundamental unit of life is cell, right? You must have heard about the cell. What is a cell? So before going on that, we will see the discovery of cell, that is how cell is discovered. As I have written on the board, first of all, the scientist whose name is Robert Hooke, Robert Hooke has observed a thin slice of cork. Now what is cork? Cork is generally present inside the tree below the bark. Bark is the outermost skin of the tree, right? Outermost skin of the stem inside the tree, and once we cut the bark we are able to get cork, right? So first time the scientist whose name is Robert Hooke, he observed the thin slice of cork with the help of self-designed microscope. Self-designed microscope, you can say a very simple microscope by using a magnifying glass, maybe, right? We don't know how he made a self-designed microscope. So by using self-designed microscope, Robert Hooke has studied or observed a thin slice of cork, and he observed that is in the section of cork a honeycomb like structure is there. Honeycomb we all have seen, right? Honeycomb like structure he observed in his self-designed microscope. Then in 1665 he has done this observation, and it is a chance discovery. Chance discovery means what? He doesn't purposefully observe that a slice of cork. He just observing it for its interest, and he discovered the structure of this honeycomb like structure. That is, this honeycomb like structure which is having a compartment. Honeycomb like structure is having compartment, and he named this compartment as a cell. Then cell word comes from the Latin language, that is Latin word, and in general if we know the meaning, if we go to know the meaning, cell means little room, right? So many little room like compartments comes together and they form a honeycomb like structure. Afterwards, this is a very small and insignificant discovery, right? Very small discovery you can say. That is when Robert Hooke was observing a slice of cork, he find out a honeycomb like structure, and in honeycomb like structure each and every compartment he gave a name as a cell, and cell means little room. If we think it is a very small and insignificant discovery, but afterwards it become a very important discovery of the biology to study the living organisms. Why and how, that we will study in this chapter, right? Now very first time someone has observed this kind of structure in the living organism. Why it becomes a very important discovery in biology? That is, very first time anyone or someone has observed this kind of structure inside the living organism, that is inside plant, and they concluded that each and every living organism is having this kind of compartment which is known as a cell. And till date this discovery is done in which year? 1665, right? Till date we are using this cell as a name of the compartment which are present inside our body or inside other organisms, right? So it becomes a very important discovery. So this is history. In that you have to remember that is how Robert Hooke has discovered a cell. Afterwards, your first topic, that is what are living organisms made up of. Once Robert Hooke discovered cell, and he said that in the plant there is a presence of compartment which are given as a name cell, right? Afterwards, other scientists and other researchers who are interested in the living world, they have started finding out new ideas, that is what are living organisms are made up of, right? Living organisms किससे बने हैं? So for that, one activity is given in your textbook, so we will discuss that activity, so some ideas will be clear in your mind. In this activity, what you have to do, you have to take a onion bulb, right? You have to take onion. So peel the onion, and you have to take a white color, very transparent, translucent type of skin from the onion on the concave side. If you peel off the onion, right? Suppose it is a onion. When you peel off from this concave side, a very thin white color layer is there. That you have to take. Take that skin, put it into the watch glass. In watch glass, take a water. So why we are doing that step? Why we are taking peel from the onion bulb and we are putting it into the watch glass containing water? That is to prevent the drying and its shrinking, right? Generally, if we very common example, if you pluck a leaf and you keep it aside, after some time it gets dry and it somewhat gets shrink, right? So prevent shrinking and drying of our cells of the onion bulb, we are putting it into the water in watch glass. Afterwards, take a glass slide. What is glass slide? It is made up of glass, right? Very small, that you will see when you come into the school, right? Glass slide, and on glass slide put a drop of water on it, and with the help of very fine paint brush, right? With the help of paint brush, take a onion peel from your watch glass and put it into the glass slide, right? Your watch glass is also made up of glass, very small dish like structure. Suppose here you have taken an onion peel. With the help of brush, you have to put it into the slide, right? Glass slide. In glass slide, you have to put a drop of water in the center and then put your onion bulb peel, right? Or you can say skin. Then afterwards, cover your skin. Okay, before covering the skin, you have to take care, that is your onion peel skin must be in the flat position, right? It must be in the flat position. Then afterwards, put a cover slip on it with the help of needle. Put a cover slip on it with the help of needle and avoid air bubbles. Otherwise, when you observe it in the microscope, you will see air bubbles mostly, right? Then after putting a cover slip, avoid air bubbles, observe your onion peel skin into the microscope. When you observe it into the microscope, you are able to see the structure which looks like a honeycomb. So many compartments are there, right? If I draw over here how it looks like, see. These are the compartment like structure which we are able to see under the microscope. Okay, observe it into the high power and then observe it into the low power and then observe it into the high power, and you are able to see this kind of structure inside your onion peel skin, which is known as cell, After doing this activity, one thing we can understand, that is your onion bulb is made up of this type of cells, right? So many cells comes together and they form a structure, that is onion bulb. So very small unit, that is a cell, they comes together and they form a large structure. It is similar for all type of organisms like us and other plants, other insects, reptiles, aves. All type of organisms are made up of single cell, right? And then that cell go for the cell division and it forms a organism. Okay, another thing, this is from the onion bulb, right? If you take onion skin from different part of the onion bulb, then what you are able to observe, अलग अलग जगह से आपने ये सेक्शन लिए हैं स्किन के ऑनियन बल्ब में से. So what you are able to observe, all the cells are similar with each other or they are dissimilar with each other. If we talk about only onion bulb, then they all are similar with each other. Structurally they are similar because they all are from the single onion bulb, right? So that is the conclusion, that is so many cells comes together, small units comes together and they form a large structure, right? Another, if you take a section from the onion roots, onion leaves, right, and you observe like this and you observe with this activity, and when you see them in the microscope, are you able to see any difference between the cells of onion bulb, cells of onion root hairs, cells of onion leaves? Then yes, that in the single plant or in the single organisms there are different type of cells, so many type of cells which are dissimilar in their size, shape and even in the function. Okay, one step is over here I have written, that is use of safranin solution. Safranin solution, it is pink in color. Why we are using safranin solution? Because generally all the cells are colorless. Our animal cells or plant cells are generally green in color because they are having chlorophyll pigment. But if we talk about animal cells and these onion peel cells, they are generally colorless. So to observe them properly in the microscope, we are staining them. To stain means to color them. So we are able to observe them properly. So by staining, for staining we are using safranin solution. So we are able to see the nucleus which are present in the cell clearly, which gets stained in the pink color. Okay, so this is the basic concept, that is how the cell discovered. It is discovered by Robert Hooke, and then you have to go for the activity, and from activity you have to understood that is every large organism is made up of very small unit, that is a cell. As I have said, in a single plant, like if we take an example of onion plant, in the onion root, in the onion bulb, in the onion leaf, if you do this activity you will find a same compartment like structure, but they all are differ from each other in size, shape and their function. So just go through it, just take its screenshot, then we will move on next topic. Okay. Okay, now students, so we are doing what are living organisms made up of, right? So in that we will go just little deeply. That is, we have discussed one activity, that is onion bulb activity. That is by performing that activity we are able to understood that onion bulb is made up of so many single compartment, that is the cell, and that small structures comes together and they form a large structure. Now we will go little deeply, that is what are these structures. The name is already given by Robert Hooke, that is these structures are known as cell. Another, these structures are similar to each other and they together they form a big structure. As I have said, right, small compartments comes together and they form a big structure. Now cell structures are basic building unit of the onion bulb. Each and every cell, when they comes together, they form a onion bulb. So they act as a building blocks, right? They act as a building blocks. If you have played that blocks when you are children, when you are a child, you have seen that by arranging the blocks you are able to make a different size, different shape buildings and other structures, right? So similar way, when cells comes together they form a different structures. Then all organisms are made up of cells. All the living organisms, either me, you, plants, animals like cow, buffalo, horse, donkey, cat, dog, all are made up of cells, right? And we all are made up of single cell. What is that single cell? That is known as zygote. If you have heard this word zygote, when male and female gamete fuse with each other, it forms a zygote, and zygotes do the cell division, increase its number, and then finally it gets converted into the living organism. Then there are some single cells also who act as a single living organism, right? They live on their own. One single cell is able to live on their own. No other cells are required to complete their life cycle. A very common example, your friend, that is amoeba, right? You heard about amoeba, right? Amoeba, paramecium, they are the single cells which act as a living organism. After the discovery of Robert Hooke, other scientists also came who has done so many discoveries in the discovery of cell and in the understanding, that is how organisms are made up of. So first one, Robert Hooke, that we have seen, he discovered a cell in the year 1665. Afterwards another scientist came whose name is Antony van Leeuwenhoek, and Antony van Leeuwenhoek, with the help of its self-made microscope, he also found a free living cells in the pond water in 1674. Then Robert Brown, scientist, came and he found nucleus in 1831. We know the nucleus. If I draw a cell over here, suppose this is a cell. Inside the cell we are able to see one dark color spot or circular spot, that is known as nucleus. So discovery of nucleus is done by Robert Brown. Afterwards another scientist named Perkinge, right? Perkinge has found out a protoplasm, that is the fluid substance which are present inside cell, in 1839. After the discovery of this, after the discovery of cell, after the discovery of living cells in the pond water, nucleus by Robert Brown, protoplasm by Perkinge, another three scientists have came and they gave a cell theory. They gave cell theory. So what is cell theory? Theory means basically related to some facts, right? So three scientists, that is Matthew Sliden, Theodor Swan and Rudolf Virchow, these three scientists have given a cell theory. So what is cell theory? They said, they gave a statement, that is cell is a structural and functional unit of each and every living organism. Cell is a structural and functional unit of each and every living organism. Second statement which is given by them, that is all the living organisms are made up of cell and cell products. All the living organisms are made up of cell and cell products. And the third statement, which is after so long, in 1855, given by Rudolf Virchow. Here you can see Sliden and Swan, they have given their statement in 1838 and 1839. After 10 to 15 years gap, Rudolf Virchow came and he added another one statement in 1855, that is all new cells comes from the pre-existing cells. All new cells comes from the pre-existing cells. So after the discovery of cell by Robert Hooke, we are able to understood, that is how living organisms are made up of, how single cells comes together and they form a large structure. Sometimes some single cells act as a single individual organisms. Another scientist came and they have done the study with the help of simple microscope and magnifying glass, and they find out many structures inside the cells, like nucleus by Robert Brown, protoplasm by Perkinge, and the three scientists Theodor Swan, Matthew Sliden and Virchow, they have given a cell theory. After this much discovery, in 1940 there is a discovery of electron microscope. Electron microscope was developed in 1940, and by using this electron microscope we are able to understand the internal structure of cell. Internal structure of cell means what is present inside the cell, which is responsible for making a large structures, which is responsible for making a living organism. So with the help of electron microscope we are able to understood that inside the cell there is a presence of organelles, cytoplasm, organelles like nucleus, mitochondria, Golgi apparatus, endoplasmic reticulum. These organelles we are able to study in detail with the help of this electron microscope. Then single cell may constitute a whole organism, as we have discussed over here. Sometimes single cell also act as a living organism on their own. That same thing is over here, that is single cell may constitute a whole organism. A single cell will act as a living organism. For example, amoeba, chlamydomonas and paramecium, and another one that is bacteria. They are the unicellular organism. So on the basis of number of cells, there are two type of organisms on earth. What are that two types? That is unicellular and multicellular organism. Unicellular organism means they are made up of single cell. Single cell will act as a living organism. Multicellular organisms means so many cells comes together, they form a large structure that are known as multicellular organism. As I have written over here, amoeba, chlamydomonas, paramecium, bacteria, they all are the unicellular organism. Apart from this, if you find out the example of unicellular organism, we can take example of desmids, then dinoflagellates, then protozoa. They are the another examples of unicellular organisms. And if we talk about multicellular organisms, we can take entire plant kingdom and animal kingdom. We can take plant kingdom and animal kingdom as a multicellular organism. Right now, multicellular organisms have come from the single cell. That single cell, as I said, that is zygote. How zygote is formed? You must have studied in your 7th and 8th. That is when gametes, male gamete and female gamete, they fuse with each other. They are able to form zygote. Zygote will go for the cell division and it produces large organism. Right? That is multicellular organisms also start their life as a single cell. Then another activity is given in your textbook, that is activity 5.2. In activity 5.1, what we have done, just recall that we have taken an onion bulb. From the onion bulb we have take a peel from the skin and we have stained it with the safranin. We observe it under microscope and we have seen so many compartments and concluded that onion bulb is made up of that single compartment known as cells. When they comes together, they form onion bulb. So same activity is over here. In this activity, you have to take different cells from the different part of the plant. That is, you can take onion peels, you can take leaf peels, you can take tip of roots, and after doing that activity, you have to answer some 4 questions to understand cell and living organisms are made up of cells properly. So first question, do all cells look alike in the term of shape and size? जो जितने भी सेल्स आपने जितने भी सेक्शंस ऑब्जर्व किए उसमें हर एक सेल आपको एक जैसा दिखा हर एक सेक्शन के अंदर? ऑल सेल्स आर सेम इन साइज एंड शेप? नो, द सेल्स आर डिफरेंट इन साइज एंड शेप. राइट? If you observe onion peel, they may be different in the size and shape. If you observe a leaf peel, that cells may be different in the size and shape, and tip of roots, that may be also different in size and shape. Another question, do all cells look alike in structure? Do all cells look alike in structure? Yes, they are similar in the structure. How? That is, they all are look like a compartment. They all are look like a small compartments. Their size and shape may be different, right? For example, your cells may be a circular, they may be oval, they may be square, they may be hexagonal, right? Different shape of cells are there. Then could we find difference among cells from the different part? Yes, we are able to find a different type of cells in the different part of the plant or different part of the animal. That is, structurally they are similar, but functionally and on the basis of size and shape they are different, right? So answer of this, that is, differ in size, shape and function, right? And last one, that is, what similarities could we find if we observe different sections in the microscope from the different part of the plant? And as we have given the answer of these 3 questions, right? All the cells are similar in the structure but they are different in shape and size. Then all the cells look alike, they are similar, they look like a box or you can say compartment, but they are having a different shape. Then all the cells, they differ from each other in their size, shape and function. And the last one, similarity. What the similarity which we are able to find in between them, that is their internal structure. All the cells are having nucleus in their internal structure. They are having ribosomes, they are having vacuoles, they are having mitochondria, endoplasmic reticulum. In short, internally they are having similarity with each other. They all possess organelles for their functioning. Okay, so I hope today you understood that is what is a cell, how it is discovered, how scientists are related with the discovery of cell, and how organisms are made up of cells. What is the basic purpose of cell, that is cells comes together and they form a large structure. So in the next lecture we will go deeply in this again. So write these notes in your notebook and see you in the next lecture.